Wolfgang, thank you for such a brilliant introduction. I hope that we'll cover many of those questions in the coming couple of days. So it's my pleasure to introduce my friend, colleague, Charlie Eaton, who's a retired hand surgeon from Florida and now heads the Dupuytren Research Group to start this session on pathophysiology.
Good morning. We are still in the discovery phase of understanding Dupuytren pathogenesis. I'm going to be presenting simplistic models of what we know and what we wish we knew, broken down into 7 sections. The current section is top left, references bottom right. I have a QR code on the final slide for you to download the references. I have no conflicts of interest. We usually compare Dupuytren biology to wound healing, but it's much more like an organ fibrosis. Biologically, it has more in common with lung or kidney fibrosis. Than with wound healing. It's a chronic disease. It starts slowly, it's self-sustaining, it's a process, not an event. The Dupuytren organ is the palmar skin, anchoring fibers, fascia, and fat. All these tissues are involved, even if they look normal. Even the nerves and sweat glands in the skin are involved. Organ fibrosis can be described in terms of 2 distinct biologies. Hot fibrosis is immune-driven. Cold fibrosis is mechanically driven. In Dupuytren, the hot fibrosis is nodules. The cold fibrosis is cords and progressive contractures. Dupuytren risk begins with DNA variants. The child of a parent with Dupuytren has a 50% chance of developing Dupuytren. Dupuytren is a 2-hit disease, genetic risk and epigenetic stress. Stresses can be from diabetes, environment, or trauma, but the biggest stress is the epigenetics of aging. These DNA processes can be a tipping point to abnormal RNA gene expression measurements. RNA abnormalities can be a tipping point to abnormal protein expression measurements. Epigenetics, RNAs, and proteins are dynamic biomarkers. Their measurements don't always agree with each other. They're affected by where specimens are from and how they're handled, but they are the key to solving the most urgent need, which is developing and rapidly testing preventive treatments. You cannot do effective research to develop preventive treatments for a chronic disease without using dynamic biomarkers to do biological staging and rapid trend analysis of therapeutic response. Molecular abnormalities can reach a tipping point to clinical hot fibrosis. Now, hot fibrosis has self-sustaining interactions of immune cells, myofibroblasts, and abnormal extracellular matrix components. This is what nodules are. Sometimes hot fibrosis leads to the tipping point of cold fibrosis that has self-sustaining interactions between myofibroblasts, extracellular matrix, and mechanical stress. This is what cords and contractures are. Progression is unpredictable. 10% of nodules resolve without any treatment. Only 25% of nodules progress to contractures severe enough to need a procedure. We know a lot about each of these compartments, but we don't know enough about the tipping points that connect them. Many DNA variants have Dupuytren correlations. It's been a huge advance to develop genetic risk scores based on these variants. The red genes on this list are near DNA variants that are highly correlated, for example, SFRP4. For genetic diseases, the more severe the phenotype, the earlier the disease manifests. For Dupuytren, higher genetic risk scores correlate with earlier onset, earlier need for treatment, and earlier recurrence. These genes have high-profile RNA abnormalities that correlate with Dupuytren. Overexpressed, underexpressed, associated with hot fibrosis, cold fibrosis, or both. This list is different than the last list, but SFRP4 is still here. It's underexpressed. These are genes of high-profile proteins associated with Dupuytren. Overexpressed, underexpressed, hot and cold. This list is different than the last 2 lists, but SFRP4 is still here, still underexpressed. Dupuytren has many dysregulated biological pathways. Each pathway is a potential source of biomarkers. Wnt signaling, for example, is high profile because it's dysregulated in all organ fibroses. SFRP4 is just one of many genes that affects Wnt signaling, and one can't make predictions based on a single gene variant. One needs a genetic risk score and a dynamic biomarker profile. Next, this is normal connective tissue. Fibronectin, proteoglycans, type 1 collagen, elastin fibers, cells, and blood vessels. But in Dupuytren disease, there's some kind of tipping point that changes this picture. Immune cells appear. Perivascular cells accumulate around small blood vessels. Type 3 collagen levels rise. Immune cells release cytokines and growth factors. Transforming growth factor beta activates myofibroblasts. Myofibroblasts release cytokines, enzymes, and collagens. There's inflammation and a lot of cell-cell interactions. This is hot fibrosis. This is the nodular proliferative stage. Myofibroblasts make collagens, they contract, and they remodel tissues. These green adhesion bodies are in the cell membrane, and they connect extracellular matrix proteins to intracellular contracting fibers. Two types of myofibroblast contractions. Microcontractions move the position of adhesions. Global contractions deform the cell and the extracellular matrix. Mechanical stress provokes contractions. Microcontractions reposition the cell surface. Global contractions pull on the matrix and create slack in collagen fibrils. Microcontractions reposition slack fibrils into loops. Extracellular enzymes cleave the loops and join the ends together. This shortens individual collagen fibrils, and the cycle begins again. This progressively shortens the matrix, adds collagen, and is sustained by mechanical stress. This is cold fibrosis. This is the involutional stage. Eventually, collagen makes the matrix so stiff it stress shields myofibroblasts, and they either die or they change phenotype. This is disease remission, the residual stage. There are Dupuytren-related abnormalities throughout the polymer skin, fat, and fascia. Dupuytren begins in the extracellular matrix along lines of mechanical stress, whether or not there's superficial fascia. For example, all these cords developed in areas where there isn't any underlying superficial fascia that's shown in green. Fascia only gets involved if it's nearby and in the same direction as the mechanical stress. Clinical disease begins in locations where there are shearing forces on the tissues, where there are attachments between structures that are perpendicular to each other. This is a transverse cross-section of a cadaver hand. In the upper right, level of the cross-section, bottom left, a side view of the same level. In the mid-palm, vertical fibers anchor the skin to the underlying pretendinous bands of the superficial palmar fascia. Further out, we get to the distal palmar crease, and here, fascia connections form a vertical column that tethers the skin straight down to the bone, just at this level. The proximal and distal boundaries of this column form stress risers, where it's common for nodules to first appear. Further out, spiral bands aren't tethered to the skin, which may be why they are not the starting point of disease, although they can be involved later. Then out in the finger, Grayson fibers connect the skin to deeper structures. These fibers are normally oblique and loose. Dorsally, we see in this cadaver section that vertical fibers attach the skin to the extensor mechanism, but only at the dorsal skin creases. These fibers are stressed by extensor tendon movement, but not by movement of the adjacent skin. Knuckle pads are the dorsal version of palm nodules. Knuckle dimples, like these, are the dorsal version of palm dimples caused by shortening of vertical fibers. Stress risers and stress shielding explain common patterns of progression. When you bend your fingers, Grayson fibers loosen as they change from oblique to perpendicular. So during grip, your finger skin is mobile. It's not tethered, no stress risers. But at the distal palmar crease, your skin is tethered, regardless of finger position. And this forms stress risers on either side. This stress can provoke a tipping point for hot fibrosis, nodules, collagen accumulation. New stress risers arise at the border between normal and stiff tissues. Finger movement only pulls on tissues distal to this, so disease advances from proximal to distal. This stiff tissue can reach a tipping point for cold fibrosis, cords, contracture. This propagates distally. Stress riser, hot fibrosis, cold fibrosis, contracture. If this causes a PIP contracture, something interesting can happen. PIP capsule contractures can block extension and stress shield some proximal phalanx cords, depending on the location. This stress shielding can be a tipping point for disease remission at the proximal phalanx level. There's also stress shielding in severe contractures. This man no longer uses these fingers. When there's no strain on the tissues, Dupuytren goes into remission. It stops before fingertips make contact. If your patient has fingertips digging into their palm, there's something else going on in addition to Dupuytren, like active flexion or spasticity. Our blood biomarker research found normal net collagen synthesis, but impaired collagen degradation. Dupuytren has enzyme-resistant collagen and impaired collagenases. Dupuytren collagen is highly cross-linked, which prevents enzyme cleavage. There's also an increase in homotrimer collagen 1, which resists cleavage because it's made of 3 collagen 1A1 strands instead of collagen 1A1 and 1A2. On the enzyme side, transforming growth factor beta reduces levels of collagenases and increases levels of collagenase inhibitors. And there are Dupuytren-related collagenase variants that are functionally impaired. There are 3 clinical consequences of impaired collagen cleavage. 1, if you can't break down collagen as fast as you make it, collagen accumulates, which is a major feature of Dupuytren. 2, you need to cleave collagen in order to shorten the matrix. So cleavage problems might be why Dupuytren progression is so slow, an average of 8 years from nodule to the first procedure. And 3, slow remodeling might explain the issue of slow healing after Dupuytren surgery. In chronic diseases, acute on chronic describes the effect of an acute inflammatory stress on top of chronic disease biology. For example, trauma on osteoarthritis. It triggers disproportionate inflammation and rapid progression of the chronic disease. With Dupuytren, skin injury triggers skin-specific wound healing pathways on top of Dupuytren-related pathways. This could explain why open Dupuytren procedures have a greater risk of prolonged postoperative inflammation compared to minimally invasive procedures with minimal skin injury. It could also explain how Dupuytren can appear suddenly after a local injury, or how after an injury, contracture can progress much faster than typical Dupuytren and then slow back down when healing has run its course. This is the overlap of acute trauma biology on top of the chronic disease biology we call Dupuytren. In summary, Dupuytren is a chronic disease. It resembles organ fibrosis. It involves tipping points we don't yet understand. We need a non-invasive biomarker panel to stage the biology, rapidly test treatments, and improve long-term outcomes. The goal for everyone here today should be to support more Dupuytren pathogenesis research. Thanks. Now, please leave this slide up for a moment so people can scan this QR code to download the bibliography. While that's up, I can take a few questions. Thank you.
Thank you, Charlie, for a tour de force. Any questions from the audience?
I can't see anything.
Everyone's just scanning your QR codes,
Yes,
I think.
Everyone's asleep.
So, Charlie, can you tell us, in terms of biomarkers, do you think the blood is the right place to be looking?
I do, and the reason is that tissue markers typically you only get at the time of doing a corrective procedure, which is late stage Dupuytren. You can't use a biopsy to stage disease. If you want to do a profile, it's not repeatable. There's a risk of having provoking disease activation by doing the biopsy. And then there are issues with tissue handling. As I mentioned, the dynamic aspects of Dupuytren change depending on whether you're looking at fresh tissue, cultured tissue, animal model, whether it's, and where in the hand it's from, whether it's from a nodule, skin overlying the nodule, fat, adjacent fascia, unaffected, visibly unaffected tissue or not. So we need a repeatable biomarker that won't adversely affect the local Dupuytren biology. And there's every reason to expect this. We know that there are circulating levels that are of different proteins that are abnormal Dupuytren matrix metalloproteinases. Our research has shown that there are a number of proteins that are dysregulated in Dupuytren compared to normal. So it's the way that chronic disease research is staged in other diseases, a non-invasive marker. So,
thank you. Paul.
Thank you very much. The prevailing question for me is, what do you need from us?
we need to change our mindset and look at opportunities that we can develop research studies to look at blood biomarkers and other dynamic biomarkers that we might be able to find. There's gene expression changes that you can find in sweat glands in Dupuytren, so there may be an option to do sweat as a biomarker. I don't know. But the main thing is a mindset change, is to stop looking at Dupuytren as something that if we only had the right procedure, we'd cure it, because that's not going to happen. It's a disease that we need to develop a preventive treatment. Here's one little study that could be done. So I mentioned the organ of Dupuytren, and organs typically have some kind of unique parenchymal cell that you only find in that organ. There's liver cells, or pulmonary alveolar cells, cardiomyocytes. So what's the unique cell in Dupuytren? Well, the areas that are affected by Dupuytren are all areas where there is glabrous skin, no hair follicles, and mechanical stress. So that's the palm of the hand, sole of the foot, the shaft of the penis. And I predict that the glabrous skin is also over the areas where the knuckle pads are. That skin is different than the other areas. No one has actually looked at that histologically to see if there's if there are hair follicles there. And the importance is that there's a wound healing model by Plicus reported in 2017 that found that hair follicles elaborated a number of cytokines, including bone morphogenic protein 4, that caused myofibroblasts in the healing wound to differentiate into fat cells. So it might be not that there's a cell that's in addition to, that's abnormal, but lack of hair follicles might involve the pathogenesis. Going out on a limb, fingernails are modified hair follicles, and you never find Dupuytren nodules distal to the germinal matrix of the nail. So if we could have somebody look at the histology of the dorsal knuckle skin, that would close the loop, and I think it would be a big step forward. So that's a little thing.